Hello guys, and welcome to another video. Today we're going to be teaching NogOps about protein synthesis and / translation. Last time we taught NogOps about transcription and how mRNA is made. Today we're going to talk about how mRNA is used or translated. So we're going to do a little recap, but before we do anything, we're going to give NogOps some glasses, but since I'm struggling with this one a little bit, so we're going to give her red glasses just to show extra strength, extra determination to learn, and to show that she's really locked in today. No, why did she move? There we go. And just a reminder, this is me teaching this to myself by teaching NogOps, so I'm also learning in the process. So this one's going to be a jittery video because I'm kind of struggling with this, but we'll get through this. Right, NogOps? We will get through this here. Boom. Alright, let's do a recap on transcription. So, yes, I spelled protein wrong. I don't really care. So, transcription. We said transcription is when DNA in the nucleus turns into RNA, and then through RNA processing becomes mRNA, which then leaves the cell. So we also talked about the nitrogenous bases. Those are going to come in handy. So we know that DNA has C, G, G, A, and T, and then RNA has C, G, A, and U instead of T. Why does my G look like that? G. So it has a T instead of a U. A U instead of a T. And these bases complement each other. Specific ones complement each other depending on if it's DNA or RNA. So in DNA, C goes with G and A goes with T. And in RNA, C also goes with G, but this time A goes with U instead of T. So if we want to transcribe this message, let's say A, C, T, G, G, C, C, T, we want to take this DNA and we want to make complementary RNA. The A is going to become a U, okay, because A goes with U. The C is going to be a G because C goes with G. And then T. Since RNA does not have a T, the T will become A. Since we're going to view the T as a U, and so it becomes an A. G becomes a C, another C, and then G, and then that T becomes an A. So this is DNA, and this is RNA. Perfect. So that's how transcription essentially happens. The process of transcription is a DNA helicase, which is an enzyme. It goes into the DNA and it unzips the double helix and it reveals the hydrogen bonds, reveals and breaks the hydrogen bonds between them. And then an RNA polymerase, which is also an enzyme, takes these exposed nitrogenous bases and joins nucleotides with covalent bonds to make an RNA transcript, which is unmodified RNA, and then it goes through a process of RNA processing where it gets a cap and tail, we remove the introns, exons are spliced together, and then it leaves it. So all this is a recap. It's all in the past video, and that's what we need to know. So at the end of transcription, we end up with mRNA. So think of DNA as a recipe book for unlimited recipes. Okay, but today you're craving cookies. Okay, you just want cookies. You don't want cookies, cake, and all that. You just want cookies. So you pick one recipe from the recipe book. So DNA is the recipe book. mRNA is the cookie recipe because we want to make cookies. Okay, so couple things that we need to know before we go into it. we need to know what an amino acid is. Amino acid. An amino acid are basically the building blocks of a protein. So you have, this is a protein, okay? And then inside of the protein, it's these little amino acids that are all like connected together. Okay? And them being connected together, they create the protein. Okay? So protein is made of amino acids. They're the building blocks of protein. Now, there's something called tRNA. tRNA. tRNA carries specific amino acids to the ribosome. This will make more sense later. I might just leave it at that for now. But tRNA, the T pretty much stands for transfer. So transfer RNA. This is basically the amino acids taxi. Okay, it brings specific amino acids to the ribosome. Now, what the freak is a ribosome? A ribosome is made of rRNA. R is obviously ribosomal RNA. Now, a ribosome is what creates the protein. It's like the kitchen. Okay, remember the cookies that we want to make? Ribosome is the kitchen. tRNA is the ingredients. mRNA is the recipe. Okay, so we got our kitchen, we got our recipe. Sorry, we got our ingredients and then we got our recipe. So rRNA is the kitchen, tRNA is the ingredients, mRNA is the recipe. And what do we need to make? Cookies, aka protein. What is it made out of? Amino acids. Alright? So, whoops. It will look something like... one more thing. There's one thing called a codon. What is a codon? A codon is a sequence of 3 nucleotides. So we know what a nucleotide is. It's a sequence of 3 nucleotides that identifies which amino acid should be positioned in the next sequence. Okay, this might not make sense right now, but it will make sense later, but keep that in mind. So a codon is a sequence of 3 nucleotides on either DNA or RNA that identifies which amino acid we need. So, we're just going to look at the structure of a tRNA right now. So a tRNA, it looks something like this. Let's do it in green. Actually, no. Let's do multicolor. We've got this part. Then you've got this part. And then down here. What the hell? And then down here, you've got something like this. Alright. This up here, this blue thing, is the amino acid binding site. So this is where that specific amino acid binds to the tRNA before it is dropped off at the ribosome. Okay? So imagine this. When you go to measure out your ingredients, you put them in a measuring cup first before putting them in the recipe. This is the measuring cup. Alright? This is the measuring cup. It's where the amino acid goes before it gets dropped off at the ribosome. Alright? This down here is called the anticodon. We just learned what a codon was. It's a sequence of 3 nucleotides on the DNA or RNA that identifies which amino acid should be positioned next. So, what is an anticodon? It is the complementary codon to the mRNA codon. So, this ensures that the amino acid is inserted into the correct place of the sequence. So later on, you'll see that actually down here, what connects to this anticodon is the codon. So it looks like something like this. So the anticodon is complementary to this, but we'll get onto this when we get into the process of actually translation. So that is a tRNA. So you got your measuring cup up here, the amino acid binding site, and then the anticodon. The anticodon, what would we describe the anticodon as? It's kind of like, trying to stick to our cookies example. It's kind of like when you look back at the recipe to make sure that this is the ingredient that should be added next. Let's just leave it at that. Okay, let's leave it at that. It will all make sense later. So how are you feeling, Nogops, right now? Do you understand what tRNA is? That's the most important thing, is tRNA for now. Okay, now we're gonna get to the big stuff, okay? Now we're gonna get into ribosomes, the kitchen, where everything is made. So ribosomes. Or they could also be called ribosomal subunits. Ribosomal subunits. So, let's talk about the structure first. So, a ribosome looks something like this. It looks like a burger or a hat. Okay, then we have something like this. All right. This right here is the A site. Okay. This right here is the P site. These two, let's label with a different color. These two are tRNA binding sites. So do you remember what tRNA is? tRNA is the ingredients. Okay, so this is where the ingredients get binded in the kitchen, okay? Remember, the ribosome is the kitchen. tRNA are the ingredients. The P and A site are where the ingredients go. Okay, so think of it as like a bowl. Okay, we're putting them in the bowl. This part right here is the large subunit. And this right here is the small subunit. Alright? This right here is the mRNA binding site. So, remember, mRNA is our recipe. Our recipe is going to come in through here. Hold on. Our recipe is going to come in through here. Alright. Then it's going to go here. It's going to get an ingredient. Remember, tRNA are the ingredients. And then slowly, it's going to grow until you have your cookies. You'll see what I mean in a second. So, A site holds the tRNA that is carrying the next amino acid. Okay? A site carries the next amino acid. Think of this as a sequence, as a factory, as a conveyor belt, okay? It's the mRNA is going to move this way or this way. Yeah, mRNA is just going to move. It doesn't really matter which way it's going to move. But this is going to be your next sequence. This is what it's currently making. So this holds the tRNA carrying the growing polypeptide. What the freak is a polypeptide? Polypeptide is basically this. This is a polypeptide. Alright? Again, more on that in a second. So this is the ribosomal subunit structure. Alright?